Hello guys, welcome back to my channel. In this tutorial, we will continue with our docking tutorial. So in the last tutorial, we have gone over about preparing our protein. Now in this tutorial, we will download 5 ligands from PubChem database and convert them to PDBQT format, which is a special AutoDock format. And this conversion from SDF format, which we will obtain from PubChem database, to PDBQT format means we will prepare our ligand. So without further ado, let's go to our browser. In the Google Chrome, we will type PubChem. So we will first download a popular ligand, which is the citric acid. And we will download this in SDF format, which is a 3D SDF format, and we will give it a proper name. For example, lig underscore 1 underscore citric acid. And we will store it in our docking folder that we have created previously. And we will specify its extension in the name itself. So we will visualize this to confirm that our ligand is in proper conformation or it's not broken, and in 3D format as well. So the ligand is in 3D format and it's and it is quite healthy. That means no breakage of ligand is present. Next, we will download the ellagic acid. Download again in 3D SDF format. Again, we will give a meaningful name, which is ligand 2 ellagic acid underscore SDF. This will help us identify that the file is of SDF format. So now we have downloaded the 5 different ligands. Now we will convert all these SDF file formats into PDB file format, because SDF file format we cannot open these file format in AutoDock tools. AutoDock tools accept only PDB and PDBQT format. Therefore, we will convert these SDF files into PDB format, and for this we will use Open Babel tool. We will open this folder in the terminal, like the command prompt in Windows, and with the dir command we will list down the present files that are present in this particular folder. So with this command you can see our 5 ligand files in SDF format are present. Now we will use the obabel command. Therefore on the terminal type obabel, then specify all the SDF file, but here we will not specify each and every name of the SDF file. We will just write star.sdf. This will match all the files that will have this extension. That means the star.sdf automatically selects all the SDF files, all the files that have .sdf extension. Next, we will write -o, which is the output format we will specify. So -o, we want the file in PDB format, therefore -o PDB. Next, we will write another tag, that is -O. Here we specify the name of the output file. So the name of the output file we will specify with the same star.pdb. For example, you can see here star.sdf, the star is placed just before the .sdf extension. So the star is currently holding this part, lig1 citric underscore acid underscore sdf. This part is stored in star. So whenever the command line enters star, it will replace this line in place of star. And if we hit enter, you can see that 5 molecules have been converted from SDF to PDB format, and the first file in the conversion is lig1 citric acid underscore sdf.pdb. So we can check for the 5 PDB files with the same star.pdb extension. For that we will write dir star pdb and hit enter. So you can see that we have got total of 6 files. One PDB file is of our protein which we have prepared in our last tutorial. Now we have done with our conversion, so we will exit the terminal by the exit command. Now we can see that these files are actually PDB files. You can see that the file is of PDB format. So the file is of PDB format. So we can check by opening the file. Yes, the ligand is intact, and and all the hydrogen atoms are also present on the ligand by default. Sometimes in conversion, the ligand molecule may break. So we need to confirm that our ligand is not broken and the molecule is intact. So we will check for other atoms as well. Yeah. So all our ligands are intact and are now present in PDB format. Now we will take all these PDB format ligands into one separate folder for simplicity. So in folder, and we will name this as PDB underscore files underscore ligand, and we will move all these files in this particular folder. And now we will convert one by one ligand into a PDB Qt format. For that we will go to AutoDock tools. Here we will again set our working directory path by going to file, preferences, set. In the startup directory we will paste our path to our present working directory, that is the present folder. We'll copy this path and we'll paste it here. We'll click on set and then dismiss this dialog box. We'll go to ligand, then we will click on input and we will click on open. Here we will change our file extension to PDB files and we will select our first ligand. Click on open. Here it will tell that the number of torsions that are present in our ligand are 9 torsions. That means 9 different poses of the ligand can be formed and each pose can be docked back into the same protein. But right now we will keep the default pose of our ligand, that is we are working with the first torsion only. Now we will again go to ligand and we will output and we will you need to provide the extension explicitly, and we will go back and check whether our pdbqt file has been formed or not. So the file has been formed and ligand structure is also intact, signifying that the pdbqt file has been properly prepared. We will again confirm the file is indeed in pdbqt. The same procedure we will follow for rest of the ligand. Now we have finished converting our pdb files to pdbqt files. Now we will delete our intermediate pdb files to avoid storing unnecessary data. So we are left behind is the pdbqt files for all of our ligands. So now we have finished our preparation of ligands, and now these all ligands are ready for docking along with our protein. So in the further tutorial, in the further part of the tutorial, we will dock this protein with all these ligands one by one. So generating um, five docking complexes, and we will visualize these docking complexes in our PyMod. So that's it for this tutorial. Let's move on to next tutorial.
गांव जाने से पहले मैं तुम तीनों को ठिकाने लगा के जाऊंगा।